Cool. I hope everyone can see the slides. So I'm really excited to be here and tell you about our work studying coding variants in cancer. As mentioned, I'm a postdoc in Professor Aviv Regev's lab, and this is work done at the Broad Institute, and I'm currently continuing my postdoc at Genentech. So the work that I'm going to talk about is the work of a wonderful team, and the experiments I'll talk about were led by JT Neal, and I worked on the analysis. And this has been a really wonderful collaboration between the Regev and Jesse Bohm's group. And if you want to read more about it, you can read the paper online. And so what drives us in our work is understanding basically what cancer mutations do. And the reason why that's important, the goal is given a patient to understand what's the treatment that's most likely to work for that patient. So the way this usually works is that doctors look at the mutations that a tumor of a specific patient has. For example, here is one of these tests from Foundation Medicine. And so what the patient gets back is a list of genes together with the specific alterations found in this patient. So for example, this patient has a mutation in EGFR. And if you look more in this report, it also tells him sort of the specific therapies that are indicated for patients with this kind of mutation, which is great. However, when you look here at the bottom, this patient also has some additional mutations in disease-relevant genes. So these are also genes that would be associated with specific treatments. However, the specific mutations that the patient had are sort of without known significance. So we don't really know if they might be driving the tumor growth and should be something to target with drugs, or whether they're bystander mutations that are just accumulating but actually not important for the tumor itself. And sort of getting a complete picture of what these variants do in these cancer genes would really help even more to find the best treatment for the specific patient. And so the dream would be then to have an atlas of variant impact or an atlas of the role of these mutations across cancer genes, such that this list here at the bottom would not be present anymore. We would just know for any mutation for current therapies which one sort of applies to it. And this is challenging because there are a lot of variants, and the technologies we have to study them are still evolving. So here's an example. Here's a cancer gene, PI3 kinase. On the x-axis is the position along the amino acid sequence of this gene. And on the y-axis, I'm showing you the number of times that we've seen mutations in this gene at each specific position. And what you can see basically is that there are a few, 2 or 2.5 hotspot locations, which accumulate across many, many different patients. And that's likely because those mutations are the impactful ones. And that's why they keep sort of getting enriched for in cancer populations. And in this particular case of this gene, again, having a mutation in it, it distinguishes the level of response to specific therapies. So it's important to know when a patient has a mutation in this gene, whether it's impactful or not. However, as you can see when looking at this, the majority of mutations are actually rare mutations that only occur in 1 or 2 different patients. And so for those patients, they have a mutation in this gene that's really important for their treatment, but they don't know whether it's sort of actionable or not. And that's the gap that we want to fill. So for each of these variants, we would like to know its function on the gene. So whether it's, for example, a loss of function mutation or a gain of function mutation, or whether it doesn't affect the role of the gene. But we would also like to know more downstream consequences of these variants, for example, on tumor fitness and ultimately on response to specific drugs. So this is the dream, and of course it's a challenging one. First of all, because there's a large space of possible variants to test. And secondly, because in order to test the role of genetic variants for genes, you have to know what the gene is doing, because you have to develop an assay that reads out the function of that gene. So whenever there's a variant in a new gene that doesn't have an assay, it takes a sort of years of work to develop an assay that specifically measures effects on a gene. And so where the field is going, and what I'll also talk about today, is thinking about ways that we can annotate the effect of many coding variants in high throughput with a single assay that can be applied to many genes. It doesn't sort of have to be recreated for each new gene. And so the way we think about it is that if we want to go from variants to these different ways of quantifying their impact, we can first do some sort of measurement that allows us to embed them in a phenotypic space where variants that are similar to each other are close in this space. And then all we have to do is figure out how different groupings in this space map ultimately to what we're interested in, which is the function of the variants. And today I'll talk about doing this, this measurement or this embedding using gene expression as a readout. But as we'll talk about, there are many other ways in which you could imagine doing this that would be really interesting. So how are we going to measure the role of genetic variants in cancer genes? So the simplest way to think about it would be to basically try to engineer the variant in cells at the endogenous locus of the gene. And that's, again, sort of the path where the field is going. And it's becoming more and more efficient to use base editing for this type of work. However, when we were working on this, base editing was not very efficient, so we came up with another way to do it, which is to think about variant function as the reaction of cells when you overexpress the variant. So this is a little bit of a new idea, so let me walk you through it. So we start with a cell, and we overexpress just the wild-type normal version of the gene we're interested in. And then just this overexpression will induce a change in the cellular state of the cells. In this particular case, I'm showing it as the cell becoming a square, but it will basically might lead to different transcriptional cascades that affect specific pathways. So then we can do the same with the variant. We can take another cell and overexpress the variant and essentially ask ourselves whether overexpressing the variant is equivalent to overexpressing the wild-type version of the gene. And if it is, then that means we cannot really distinguish the effects of the variant from the wild-type. And that's what would happen if the variant doesn't interfere at all with the function of the gene. Another option of what could happen is that we overexpress the variant and it looks like we didn't overexpress anything. It just looks like the original cell. And that's what you would expect when there's a loss of function, when there's basically like a non-functional protein that won't do anything. And the third option is that something new happens to the cell that's different from when you overexpress the wild type. It's also different from what the cell used to be, and that would be a change of function. For example, a gain of function, or a dominant negative, or any sort of more nuanced effects that variants can have. So basically what we're doing is we're using cells as biosensors. We're using them as sort of math that can tell us this variant is equal to wild type or it's not. And if it's not, we can try to think more about why, in which way it's different. So the way we measure these changes in cell state, the way this has been done in the past was using bulk RNA sequencing for each of these conditions. So then again, neutral variants, when we look at their gene expression profile, would look identical to the cells that overexpress the wild type. And then cells with a loss of function would not have many gene expression changes, and those with a change of function would just have a really different one compared to what the wild type does. So this type of approach was called EVIP, for expression-based variant impact phenotyping, and it has been used successfully across a variety of different cancer genes, from tumor suppressors to oncogenes and so on, kind of showing that this would be one way in which to quantify the effect of variants across genes that have wildly different functions. So as long as they produce a change in gene expression that we can measure, then this type of assay would work. And so the contribution of the work I'm going to talk about is taking this type of approach and scaling it up using single-cell technologies. Basically going from bulk, where you have to do each condition individually, to being able to do this type of experiment in a pool and study hundreds or thousands of variants at once. So the way we do it is we start with the coding sequence of the different variants we want to study. So each variant has basically a mutation at a different position in the gene. And we associate each mutation with a specific barcode at the end of the gene. And then we take this library of variants and transduce it into cells, select for the cells that have at least one variant, and then do a single-cell RNA sequencing. And that gives us both the gene expression profile for each cell, but also the identity of the variant that was overexpressed. And then from this experiment, we can basically backtrack and take all the cells that have a specific variant and compare what they look like with the variants that were overexpressing the wild type. So this type of approach would look something like this. So when we look at single cells, we can display them in this low-dimensional representation called UMAP, where basically each cell is a dot, and cells that are similar to each other in their gene expression profiles are close to each other in this space. And so here I'm showing you the density of these dots for different conditions. So for example, in the top left here, we have in gray the wild type sort of overexpression and the sort of the gene expression space that the cells fill. And variants that are wild type like, so variants that are sort of neutral and don't do anything, will look identical to the wild type overexpression. And on the other hand, the loss of function variants and the change of function variants will stop overlapping as much with the wild type and occupy a different space here. And again, as I mentioned, the way we distinguish the loss of function from the change of function is by comparing to cells that are unassigned or they don't have any variant in them. And then the loss of function variants would be super similar to these unassigned cells, whereas the change of function would be different from both the wild type and the unassigned cells. So through this, you can kind of see that variants with different functionalities would produce differences in gene expression profiles that would allow us to categorize each variant based on its effect. So this type of approach addresses some of the challenges I mentioned in the beginning. First of all, by being compatible with pooled perturbations, it means that it's scalable and start allowing us to cover more of the space of variants. The second really important one is that it's gene agnostic, so it can be applied to variants in any gene as long as overexpressing them produces a gene expression change. And the bonus here is that we can actually look at the gene expression profile and get a lot of information. So for variants that don't look like the wild type variants, we can categorize them based on exactly what pathways they induce or repress. So with that, I'm going to talk about a proof of concept experiment we did in our paper, studying 200 variants in two important cancer genes, the tumor suppressor p53 and an oncogene KRAS. And these were chosen because they have been studied before with other assays, so we could compare our method, but also learn maybe something new about them. And so for each of these two genes, we chose the 75 most frequently occurring mutations in cancer as our test set. And then we had some controls that included synonymous variants, as well as missense variants that were observed in the general population in the exact database, but not in cancer. And those would be expected to not have strong effects. And we did this in the A549 cell line, which is a lung cancer cell line, and we used 1,000 cells per variant in order to make sure we can capture even small effects of the variants. So what did we find from this work? So for p53, I'll start that. We, of course, did a lot of quality checks to make sure that variant barcodes are expressed at similar levels, as well as that we're confident in our annotations of which cell had which variants in it. And then the first thing we need to do is figure out how to quantify the amount of, basically the effect size of a variant. How different is it from the wild type? And for that, we use the Hotelling t-square statistic in principal component space. So essentially, we do principal component analysis on all the cells in our data, and then we want to compare cells with the variant with the cells with the wild type overexpressed. And we do that comparison by comparing their principal component representations for each principal component. We essentially do a t-test, and we combine them together into this t-square statistic. And a large score is basically the more significant difference between the variant and the wild type. And then we can sort of get a significance of this by estimating empirical p-values from comparisons between synonymous controls and doing multiple testing correction by q-values. So this is what the scores look like for p53 variants. Excitingly, the controls we had, synonymous and exact, fell below the cutoff of 5% FDR, whereas the hotspot variants and the rest of the variants that we chose to be enriched in patients had high scores as we expected. But we can learn much more about variants than just how impactful they are, because we have these rich gene expression profiles that can teach us about sort of variant classes. I'll talk about that in a second, but I will say that our scores were very concordant with specialized p53 functional assays that were developed for this specific gene. When we look at the gene expression level, sort of learn more about the variants, this is what it looks like. So each column here is a gene and each row is a variant. And so for each variant, I'm showing you the average gene expression profile. And you can basically see there are two main groups. The first one contains all the control variants, so we call those ones the wild type-like. And the rest include the hotspot variants, so they are the impactful ones. And we did comparisons with specific signature genes that are expected to be induced by functional p53, so by the controls. And indeed, that's what happens, and it doesn't happen in the impactful sort of loss of function p53 variants, and that's true for a bunch of different signatures. And finally, we looked at how variants differ at the single cell level. What actually happens in terms of the cellular state distribution. So for that, here I'm showing you again the cells in the low-dimensional representation in UMAP space. And I'm showing you the density of cells that either have wild-type-like variants or impactful ones. And essentially, it looks like the cell state at the top is lost from wild-type-like to impactful variants. And that actually corresponds to cells that are non-cycling. So when we look at the cell cycle phases of each cell, we find that basically the difference between the impactful and non-impactful p53 variants is that the wild-type-like variants induce this non-cycling enrichment, which is what you would expect, because overexpressing a functional p53 would lead to cell cycle arrest. So in summary, for p53, I've told you that our approach distinguishes control synonymous p53 variants from the impactful ones through gene expression measurements, and that our approach agrees very well with other assays that were specifically developed for p53. And that when we look at the actual gene expression profiles, p53 variants are marked by a change in cell cycle progression. And while this is the case for p53, we'll see that for KRAS, the differences in gene expression between impactful variants and neutral ones are not related to cell cycle. So it's not that the only thing that we can capture is cell cycle. So I'll briefly tell you about KRAS in the last few minutes. Again, we did a lot of quality control to make sure that variants were expressed at similar levels. And again, we computed these impact scores or effect sizes of the variants. And here in gray are the variants that were controls, and most of them were, again, falling below the 5% FTR line. And the rest of them are impactful. So when we look at the gene expression profiles for KRAS, compared to p53, there are more than just two groups. So there's at the top the group of wild-type-like variants, which includes variants that are similar to when we just overexpressed the wild-type version of KRAS. But then there are four other groups of variants, each with different gene expression profiles, different pathways that are activated or repressed. And the last one is the one we call gain of function, because it includes most variants at positions 12, 13, and 61, which are sort of bona fide known gain of function KRAS variants. And the other groups, we've sort of extensively studied what these different gene expression profiles are, and you're welcome to look in the paper more. Again, our results agree quite well with an assay developed specifically for KRAS, which measures growth in low attachment. So cells can only grow in this low attachment environment when they have a KRAS gain of function variant. And indeed, the highest scores for this assay are in the gain of function group, and no significant score is in the wild-type-like group. So that gives us confidence that this assay can work both for a tumor suppressor like p53 and for an oncogene like KRAS. What was particularly interesting in the KRAS case is that the more we looked at these expression profiles, we started realizing that these groups are not necessarily completely discrete and distinct, but rather they seem to form a sort of a progression, both between groups and also within the gain-of-function variants. And you can see that really well by looking at the principal component 3, which was the one that was enriched in the gain-of-function variants. And specifically the wild-type variants, their distribution for these scores are right on top of the gray one, which is the one for the wild-type overexpression. But as you go to impactful 2, 3, 4, these distributions deviate in a sort of continuous fashion. And moreover, we see this type of continuous level of gain of function in the cellular assay that I just mentioned, the growth and low attachment, even though that assay was done in a different cell line. So this is sort of replicable across different cellular contexts. And it's not explained by just variation in the level of the variant overexpression. So the way we think about it is that there's a cell state space and variants are sampling from this space, with wild-type variants sampling more from the wild-type-like state, so in this case it's more gray, and weak gain-of-function sampling more from the middle, and the strong gain-of-function sampling from the most sort of gain-of-function state. But all of the variants having a mix of all these states, but just the proportions are different between them. And this is actually what we see in our data. So I'm showing you here the cells in the low-dimensional representation in UMAP space, with cell state 0 being the one that's enriched in gain-of-function variants. So now I'll show you how different variants are distributed in this space. Right here. So in dark red is the distribution of a known gain-of-function variant, which is right on top of the cell state 0. In black are the wild-type overexpressing cells. And in pink here is a synonymous variant. So this one overlaps completely with the wild-type overexpressing cells because it doesn't do anything. But as we move along the continuum of KRAS gain-of-function, there is more and more of a transition from this wild-type-like gain-of-function sort of state to the gain-of-function one. And this is actually what correlates with the functional assay that I mentioned before. So with that, I'll go to my summary. So I've told you that our approach, just like for p53, can distinguish our control synonymous KRAS variants from gain-of-function ones, that we identify 5 groups of impactful variants in this gene, and that at least in this specific cellular system, we see a continuous degree of KRAS gain-of-function amount. So it sort of adds nuance to the idea that variants can either be driver variants or bystanders, suggesting that, again, in this specific cell system, there seems to be more of a gradation and continuum between these two. And it would be really interesting to see more work to study this in increasingly patient-relevant samples. And of course, we did subsampling to see how many cells we would need to generalize this, but I'm running out of time, so I'll just skip to my summary. So I mentioned here that we use gene expression to measure variant impact, and that using single-cell technologies allows us to basically scale up these types of experiment to hundreds of variants in our paper, but in theory could be to thousands or hundreds of thousands of variants. And our proof of concept experiment that I've talked about studied 200 variants, and we correctly identified different types of variant impact, like gain of function and loss of function, and specific gene expression changes associated with these. I've also shown you that our predictions agree with the more labor-intensive and functional assays specifically defined for genes, and that looking at the single-cell resolution, looking at sort of the distribution of cell states, reveals how such variants can shift the balance between cell states. And as future directions, will be really cool to look at variant impact across multiple contexts, for example, different tissue types or different genetic backgrounds. The second one is thinking about other ways besides gene expression to profile the effects of a variant on cell state. One of the particularly interesting ones would be imaging. And then the third one, of course, is using these powerful base editing methods to now actually engineer the variants at their endogenous locus rather than relying on this type of overexpression system. And that would be really cool because it will allow us to look at interactions between variants in different genes, for example, or interactions between variants in the same gene, and also sort of look at more complicated samples and start looking at the effects of variants on microenvironments and so on. So with that, I'll acknowledge again this wonderful team, and I'll take any questions. Should I stop sharing?
Okay, thank you very much, Oana. I think you can hear me.
Yes.
Okay. There is, I think, a first question from the chat. Let's see if we have also. Okay, there are one from the Q&A that will be recorded, so I will do this one that is a little bit shorter, and then for the chat. Okay. Does the continuous gradient make it difficult to identify and/or quantify threshold to categorization?
So that's an interesting question. So I think you could think of, if you really want to categorize them as discrete, sort of finding a threshold based on what we currently know about the strongest gain of function versus the weakest ones, or for example, by comparing with the cellular assay for KRAS, yeah, just sort of find a threshold there. But I think ultimately looking in patient samples and seeing at what threshold you really start seeing more phenotypic changes in terms of growth and things like that would be a good way to do it.
Okay. So from the chat, among the, I think is more than one. Actually, there are 4 chats. I think is the person Antonio. Okay. Actually, I don't know if the people in the audience can actually speak up. But yeah, let me read because there are 4 questions. I don't know if we have time to answer to all the questions. Among all the different type of scenarios observed through the expression-based variant phenotyping, do you distinguish which cases come with presence absence of different expression of the target gene itself? Yeah, this is actually an interesting question. So how much you can, if I understood well, the question is how much you can differentiate between the effect of the variants that you are studying and all the possible interaction between other variants that are present.
So if I understand the question correctly, it's if there were a change in the expression of the gene itself, then how would that affect our sort of categorization of it? And so far in our work, that didn't happen. So most of the variants and the underlying genes were expressed at similar levels. And so I think for the variant sequences being expressed at different levels might mean you have different ability to detect them. So you might detect fewer cells that have a variant that makes the gene less expressed. And yeah, it would be interesting to think about ways to go around that.
Okay, the second question is, in the Perturb-seq data analysis, how can you discriminate batch effect correction from actual sample level differences?
So yeah, so this is actually one of the really cool things about Perturb-seq is that you do this experiment in a pool. So you have all the variants sort of combined in the same batch. So in our case, the batches were the different 10x channels, so the different sort of libraries that we made. And each of them contained all the variants, and we regressed out batch from the gene expression data, and yeah, basically remained with.
Okay, let's see how much. So maybe we'll have time for another question because here there are many. So let's do the third one. Do differences on single cell expression looked after matching cell by the expression level of the target gene or regardless of the later?
So as I mentioned, that the expression of the target gene was pretty constant across our cells, so there were not many gene expression changes related to that.
Okay, let's do the last one. Synonymous variants were used as control. However, some have actually have said, this is, I think, an important question, have regulatory consensus. Can you comment on whether you found interesting findings here?
Yeah, so I think, I don't know, in the study in particular, the synonymous variants were quite similar to each other, so we didn't find anything that was of a large magnitude, that was different. But yeah, I've seen other papers looking at this in more detail, so maybe the changes are small and we were not powered to detect them. Yeah.